This is the wrong one. Hereby, I open this academic ceremony in which Ms. Chang Lu will defend the academic thesis entitled Computational Strategies in Cardiometabolic Diseases, a Portal to Deeper Mechanistic Understanding. May I invite you, Ms. Lu, to present a summary of your studies in the next 15 minutes and also end with the conclusions of your thesis. I'd like to give you the word.
Dear Prorector, members of Corona, family, friends, and colleagues, welcome to my defense. Let me first introduce you one of the biggest public health problems of the modern time, cardiometabolic diseases. Cardiometabolic diseases form the largest contributor to the mortality worldwide. They cause around 1/3 of deaths per year, not only in Western society, but also in emerging economies. They are also a major cause of morbidity, which comes with the hospitalization and the long-term medication use. Therefore, they cost our society a lot of money. When I talk about cardiometabolic diseases, I refer to a cluster of nutrition-related disorders, including cardiovascular disease, such as ischemic stroke and coronary artery disease, plus others such as non-alcohol fatty liver disease. So why do we see so many cardiometabolic disease patients around the world? Well, they are mainly caused by our modern lifestyle, such as smoking, unhealthy eating, and lack of exercise. Other factors, including age and sex, also influence their development and progression. Among these, atherosclerosis is a pathological basis of the coronary artery disease and stroke, which are two of the most dangerous cardiovascular diseases. Atherosclerosis is a build-up of fat, cholesterol, and other substances in and on the aorta wall. This build-up is called plaque. Monocyte-derived macrophages are produced in response to inflammation and stewed into the plaque, where they take up lipid particles and transform into foam cells. Macrophages adapt to stimuli in their microenvironment to adapt to a spectrum of functional phenotypes. Therefore, it is an urgent need to develop effective methods to understand the macrophage's heterogeneity in its microenvironments. Multiple high-throughput technologies and computational methods allow us to measure molecular profile at a single-cell resolution, including genes, proteins, and metabolites, and then identify cell phenotypes or analyze the molecular contexts. However, to study cells' microenvironments, we still need cells' locations on the tissues to assess their spatial distribution. Therefore, in Chapter 2, we designed a pipeline to integrate multispectral and mass spectrometry imaging to map macrophages' heterogeneity in its molecular and cellular context. We cut plaque sections from LDLR receptor knockout mice fat high-fat diet, and then adjacent section was scanned by different image technologies. Here, the upper one was analyzed by multispectral imaging microscopy to visualize up to 13 biomarkers. After cell segmentation, we clustered these cell segments into 17 phenotypic clusters based on the biomarker's intensity per cell. The lower section was analyzed by mass spectrometry imaging to visualize their lipidomic profile. Here, M over Z means mass to charge ratio, standing for the molecular weights of the sample component. By aligning to adjacent sections, the cell phenotype defined from the upper layer can be mapped to the lower layer. Thus, each pixel of mass spectrometry imaging can be labeled as a certain phenotypic population. Finally, we extract characteristic lipid signatures for each phenotype. Here is an example. Cluster red, yellow, and blue were identified as adventitial phenotypes according to their intensity of biomarkers and the spatial localization on the tissues. By linking these clusters to their corresponding multi-MSI data, characteristic MD values are able to distinguish the phenotypes well, which proves the effectiveness of our model. In addition to spatial information, temporal information is also important for analyzing the mechanism of cardiometabolic diseases. For example, non-alcohol fatty liver disease may further progress to NASH and subsequently to cirrhosis and even cancer. Many studies have demonstrated the metabolic disarray in the liver tissue during the disease progression. However, it is still unclear the trajectory of metabolites with the progression, especially in individual liver compartments such as parenchyma and the sinusoids. Therefore, in Chapter 3, we took advantage of both spatial and temporal information to analyze metabolic changes in the early stage of non-alcohol fatty liver disease at the global and compartment levels. We cut liver sections from 27 LDL receptor knockout mice fed a high-fat diet for 0 to 3 weeks, and then generated 27 multi-MSI data cubes, where the z-axis is mass-to-charge ratio, standing for potential metabolites on the tissue. The MD values were then clustered into several metabolic clusters based on the time course of the average intensity of all pixels in each section. Subsequently, we developed a strategy to annotate MSI pixels to a particular liver compartment, results in the accurate identification of 3 major liver compartments, which are parenchyma, sinusoid, and vessel. These compartments are independent with the disease stage. Finally, based on these annotations, we mapped metabolites clusters within the parenchyma and the sinusoid compartment to reveal unique dynamic changes with the disease progression. Our pipeline is able to distinguish liver compartment based on metabolite signatures from mass spectrometry imaging without relying on the annotation from the histological image, indicating the presence of metabolites, describing liver compartment heterogeneity. Furthermore, temporal analysis on parenchyma and the sinusoid reveals their differences in the temporal response to high-fat diet. As we mentioned before, the sex difference in cardiovascular disease is well known. But can this difference be explained by our genes? And what are the potential signaling pathways contributing to this difference? These are the questions we studied in Chapter 4. First, we generated sex-based gene expression signatures by comparing male and female patients' gene expressions. These signatures told us which genes are significantly differentially expressed between male and female patients. Comparing the information from prior knowledge resources, we use multiple computational methods to infer which signaling pathway were associated with genetic difference. We also built a cytokine transcription factor network by integrating the prior knowledge resource and the coronary artery disease cohort to investigate whether common regulatory mechanisms underlie the sex-specific pathway activation. Next, after inspecting the cytokine activity and the differential gene expression in an independent healthy cohort, we confirmed the specificity of these sex differences for coronary artery disease. The risk factor of cardiovascular disease has been linked not only sex, but also a number of biological factors. In Chapter 5, we studied the impact of cardiovascular disease risk factor on monocytes response, in particular in infectious stimuli. Therefore, we compared the monocytes gene expression profile from coronary artery disease patients before and after LPS stimulation, results in an LPS response matrix. By calculating the correlation between the LPS response and the risk factors, we found the patients with high blood pressure show a weaker LPS response than those with low blood pressure. This suggesting high blood pressure in coronary artery disease dampens the response of monocytes to LPS. Next, we generated a gene co-regulation network and identified the gene modules that shows a strong correlation to blood pressure levels is related to oxidative phosphorylation. We also built a gene regulatory network and inferred crucial transcription factors such as ZBTB43, because they are not only drive the most of hub genes in this module, but also have the highest transcription factor activity. Finally, we screened potential drugs that can reverse this weakened LPS response. We identified iloprost, the drug for reducing the pulmonary artery hypertension, increase the LPS response of monocytes, hereby validating our results. In conclusion, in this thesis, we analyzed the multiple spectral and temporal high-dimensional omics data through several computational strategies to dissect the heterogeneity of macrophages in atherosclerosis and the progression of non-alcohol fatty liver disease, and the risk factors of cardiovascular disease before and after infections at the spatial and temporal or molecular levels. These findings, based on this analysis, deepen the understanding of cardiometabolic diseases and help us develop more effective treatment options to cure the disease as early as possible. Thanks for your attention. Now I give my word back to the prorector.
Thank you very much, Ms. Candidate, for your clear presentation, well in time and well illustrated. My compliments. We now will continue with give you the possibility to defend your thesis, and we have 6 opponents. And for the audience, we have 5 opponents who are present here on site, and we have 1 opponent who is online present here. I would like to start giving the possibility to start the opposition to Professor Jankowski. He is both affiliated with Maastricht University as well as with the RWTH Aachen in Germany, and he has an expertise in proteomics, and he was also a member of your assessment committee. And I'd like to give you the first word.
Yeah, thank you very much. Yeah, first of all, dear candidate, I want to congratulate you for this thesis. It was really well written and impressive, and it was a pleasure to read this thesis, first of all. In your introduction of your thesis, you mentioned that there are a lot of studies have been identified that vascular cells produce a response to inflammatory cytokines. My first question is, could you explain how during an inflammation process cytokines recruit leukocytes and monocytes on the inflammatory side, and which immune processes are involved in this recruitment?
Highly esteemed opponent, thanks for your kind word and question. Yeah, so I'm sorry, could you repeat it again? So,
Yeah,
a little bit slowly.
Could you explain the mechanism, how cytokines attract leukocytes and monocytes? How is the mechanism by which these cytokines attract the cells?
Thanks for your question. If I understand correctly, your question is how the cytokines influence the monocytes.
Yes.
Yeah. So first of all, I have to say I'm not a biologist. So based on my limited knowledge and the results I got from the chapter 4, we found some cytokines are activated in female rather than in male in cardiovascular disease patients, such as GM-CSF. And for the rest of cytokines, I have to say, I didn't study that too much.
Okay, thank you.
Thank you.
In your thesis, you use different methods. If I understand this correctly, the methods were such as MNET and R-race. I hope I pronounce this correct. The different methods you used. My question is: are there any further methods available, or why you use these biostatistical method in the thesis? Is this the only methods which are available, or do you know some other methods to use? And why you choose these biostatistical method and not?
Thanks for your question. So could you repeat the method's name? I couldn't.
MRNet and RA-racen.
Racen.
Or why? Can you explain in general which biostatistical method you use in your thesis, and why you choose these and not others?
Okay. Thanks for your question. So the method I use always depend on what kind of data I process. So in my thesis, I produce the microarray data. For that one, if I would like to investigate the cells and the gene and the gene's relationship, I would use some graph-based methods such as WGCNA. And for the, if I would like to compare the two groups, such as male versus female or LPS versus non-LPS, then I would like to use some method like lemma. And for the also analysis some mass spectrometry imaging. For that one, actually, what we are interested in, one is we are interested in their spatial localization. And another thing is we are interested in the characteristic monocytes, our lipids, which are specific in a region or in a cell type. So for this one, I could use something like a feature selection method, or like what I use in the chapter 3, which is the shrinking centroid clustering method to identify the different regions such as the parenchyma and the sinusoid.
Okay,
Thanks
thank
very
you.
much.
My third question is regarding this multi-imaging.
Mm-hmm.
You show very impressive images within your presentation. Could you explain what we see on these images? These green and red dots. What is shown on these images?
Thanks for your question. If you see, I assume you discuss is about the liver study. The imaging you see, including pink and red and yellow colors, they indicate different liver compartment. So in that paper, actually, we identified liver compartment without histological imaging. So normally for a pathologist, if they would like to identify the liver compartment, they need to manually annotate which region is sinusoid or parenchyma. But for our method, we can do it without the histological image, only based on the mass spectrometry imaging, which will avoid the registration between the mass spectrometry imaging and the histological imaging. That is what we did in the chapter 3.
Okay, thank you. So time for one last short question. Can you explain a little bit how this mass spectrometric imaging works, the technique?
So before that, I have to see, I didn't participated in this experiment, but we collaborate with M4I, which is the Institute for the Mass Spectrometry Imaging. They did this experiment for us. And I could explain it a little bit. Is for a tissue, they scan by a technique called mass spectrometry imaging, and the laser will shoot on this tissue, and the molecular will come out, including some lipids or metabolites. And then these lipids and metabolites have different weight, and they will, in that machine, this molecular will fly to like a board or something, and then that machine will calculate the time. So for some molecular, they are heavy, then they fly slow. For some molecular, they are smaller, they fly fast. So based on this approach, they will know what kind of molecular they are. So this is a basic principle of the mass spectrometry image.
Okay, thank you.
Thank you very much. The opposition will now be continued online. We go to Professor Ulas. He is from the Department of Bioinformatics at the Institute of Bonn in Germany, and I would like to welcome him all the way from Bonn. Okay, the word is yours.
Correct. Thank you very much for your nice introduction. And dear Ms. Lu, congratulations for your very nice work and thesis, and for your very nice presentation and summary of your work. My first question is going into the chapter 5, where you describe gene co-regulation networks and gene regulatory networks. Can you maybe describe the difference of those two network approaches and how you construct those, please?
Esteemed opponent, thanks for your question. In chapter 5, we use two approaches. One is WGCNA and another is Rankina. So to generate the gene regulatory network and gene regulation network. So for the gene regulation network, actually, we are going to check the relationship between genes. So what we did is we just, the principle of WGCN is they calculate the Pearson correlation between genes, and then they cluster the genes with the similar functions or similar gene expressions. And for this, we could investigate the relationship between genes. And for the gene regulatory network, actually, we would like to check which transcription factors drives this gene's expression. So that is genes and transcription factor. So that's the difference.
Thank you very much. My second question goes into a similar direction, where you in chapter 5, you have selected in the Salmon or performed the hub gene detection in the Salmon module. I think you've used the centrality measurement to identify hub genes. Are you aware of even other matrices to measure the importance of a gene in such a network?
Thanks for your question. So for the So for your question, we in our chapter 5 we use the degree of the network to define the hub genes. So I have to say I didn't know other methods for calculate the hub genes, but what I can come up with is we can also consider the weight between the genes. If we plus the weights, then we could find maybe some important genes, not only based on the degree.
Mm-hmm. And have you heard about something like shortest path and similar things that can be also applied to networks? So can you imagine those can be also used to maybe provide even more or informative hub genes?
Sorry, I either didn't hear that name, or I don't know that that tool or.
Don't worry. So you can calculate the shortest path in a network. So how much nodes a gene has to pass until it reaches, for example, the border of the network. So this can be also a measurements for importance of a gene, of a hub gene.
Thanks for your question. I totally agree with you. So I didn't read any papers about this shortest path, but I know some methods to from the upstream gene expression to infer from the downstream gene expression to infer the upstream proteins or metabolites. They also use the shortest path to find the pathway. So that should be similar.
Exactly. Yes, thank you very much. My next question goes into chapter 4, is a more maybe biological one, where you compare the sex differences. Have you also maybe looked into whether you have maybe subgroups in the female part of your data sets, because knowing that the menstrual cycle has influence on the immune space in the blood, so this might have also an influence. So my question is, did you look also into the differences in the female cohort?
Sorry, could you repeat your question? I mean the subcohort I extracted is based on their ages. So is that what you see?
Not directly. So I'm almost sure that in this cohort, the menstrual cycle of the women wasn't tracked. So it is known that depending in which phase the women are during the menstrual cycle, the immune activation of the immune cells in the blood are a little bit different. So you have compared female versus male, but I wanted to know if you saw maybe some subclustering, if you look, for example, in the PSA or hierarchical clustering, if you saw some substructures in the female cohort which could hint towards differences coming from, for example, the menstrual cycle.
Thanks for your question. So, so actually, I didn't check if there is a subsize in a female or in the male, but I would like to zoom into it afterwards.
Okay, thank you very much. Is there time for another question?
There's no time for a further question. This was your time. So we have to continue, and we continue the rest here with on-site questionings. So the next opponent who's going to address the questions is Professor Welbig. She's affiliated with the Department of Advanced Computing Science of Maastricht University, and I would like to give you the word.
Thank you. Thank you, Mr. Prorector. Esteemed doctoral candidate, dear Mrs. Lu, it was a pleasure to read your thesis. I compliment you with the good work, especially the extensive description of the experiments. I'd admire that you could cut the mice. Despite this good impression your work makes, I'm still left with some questions I want to discuss with you. And the first one is about overarching framework. Is there an overarching framework? Because you are analyzing different diseases, different techniques. So can you combine those techniques into one framework?
Thanks for your highly esteemed opponent. Thanks for your question. To be honest, it is not that easy to combine all the functions into one framework, because different biologists or pathologists, they always have different purpose. They would like to study different. Some people, they would like to study genes. Some people, they would like to study samples, or somebody, they would like to segment images. So according to different purpose, we always need to use different methods to deal that problem. So, but there are some functions or some purpose they can combine with each other. For example, if we study no matter which disease, we are always interested in to study the single cells cell type. So for that one, people could use like a single cell analysis, such as clustering or feature extraction, to analyze the cell's heterogeneity. Or for no matter for cancers or cardiovascular disease, some people would like to study the spatial distribution on the tissues. For that one, we could also use the same approach to do the image segmentation. Yeah, that's my answer. Thank you.
Okay, thank you. And the second question is about correlations and causations. We know that machine learning and statistics is based on correlations, but causations is a completely different animal. For instance, there is a strong correlation between number of movies that Nicolas Cage was performing and number of pool drownings. But obviously we know that even if we forbid Nicolas Cage to perform, then still we will not solve the problem of pool drownings. Therefore, I'm curious, I'm wondering how significant are the patterns that you have found in your studies?
So you mentioned about the, thanks for your question. So you were, if I understand correctly, so you mean how confident for me for the results of my study. So yeah, thanks for your question. I have to say, yeah. So for the computational methods, people, no matter for the statistical methods, we could use hypothesis testing to test our results. Or for the machine learning, we could use an independent testing data to validate our results. But even for this, if I collaborate with biologists, I still need experiment to validate our results. So I think the final step is always experimental validation. So that's why you see in the chapter 4, actually, we validate on another independent cohort. And for the chapter 3, actually, we are doing some experiments for validating the liver component we selected. Yeah, thanks.
Okay, thank you. Do I have time for one short question?
Yes, you have.
The last question I have is, you are saying that you are using some advanced machine learning techniques, and I was wondering what makes machine learning or a technique advanced in your opinion?
Yeah, thanks for your question. The machine learning algorithm I use, such as in the chapter 3, I use a clustering method called the spatial shrinking centroid clustering method. And in the chapter 2, I compare the adverbs and the random forest to do the feature selection. So these are the several machine learning methods I applied on the thesis. That's my question.
Okay, thank you.
Thank you very much. Then the opposition will be continued by Dr. Seiler. He is affiliated with the Department also of Advanced Computing Sciences at Maastricht University, and in addition, he was also a member of your assessment committee.
Thank you. Dear candidate, thanks so much for this thesis and for sharing your work with us. I learned a lot reading it, and I have some questions for you that I'm just curious about. They're really hard questions in general, so there's not one clear answer to me, but I would be curious to know your opinion about it. And the first one is in chapter 2, where you worked on you used the spillover compensation method to compensate spillover in your data, and I work a lot on that. So I'm very curious. You had two methods. You created a new one, right? And then you compared it to an older method, and you concluded that there is a difference and that your method works a bit better, or at least differently. So I was wondering, how did you compare the two, and how can you kind of know that which one works better than the other? It's tricky for me.
Esteemed opponent, thanks for your kind word and question. In chapter 2, we did the cell component, and we modified a little bit of that method. The reason is, in that paper, I assume the method for cell segmentation in that paper is different from our paper. So the cell shapes is a little bit different. So if I applied our method or our cell segments using that method, then we will lose a lot of cells. So a lot of cells intensity will decrease to 0. And after biologist inspection, they think that it shouldn't like this. So I modified this method, and then we got actually more cell segments, validated cell segments. So that's why we replaced that method to ours. Thank you.
Thank you very much. I have another question to chapter 5, and it's related to a previous question about correlation and causation. It's always, causation is the big term, right? We would always want to know what causes what. And you applied an R package there, BN Learn, that I'm not familiar with. And I don't know if it's one small part of your thesis, but do you happen to know what the underlying assumptions are that makes this package learn the causal relationship between variables in addition to the correlations? There must be some additional assumptions that are being made.
Thanks for your question. For that that package, I recall they use first for that package, they assume there are some nodes or some features. They are at the start point of the Bayes network, and some nodes, they are on the end point. This is already predefined. And then they use the hill climbing methods to infer the path from the bottom nodes to the upper nodes. So that's how they generated this Bayes network.
Thank you very much. Do I have time for one more?
You even have time for 2 more.
Wow, what a luxury! So I only have one more left, so I will stretch it out. No, the last question was in chapter 3, where I really like this. You use Dirichlet processes and Gaussian processes to do clustering on spatiotemporal data. This is really I haven't seen this kind of methods applied in the real world very much, so that's really congratulations on that. I was wondering, because they're non-parametric methods, so you don't have to predefine the number of clusters, but you still have to define some hyperparameters a little bit, some way, right? I was wondering how sensitive those are to the clustering that you found, if you ran any simulation studies or anything like that.
I think I use GIPS sampling to do the define the parameters of the Dirichlet process, and actually I didn't check the sensitivity of this, because that package has an approach to automatically find the best parameters. So yeah. So that's why I didn't look into all the parameters. Yeah, thanks for your question.
All right, thank you very much for your answers.
Okay, thank you very much. Then the opposition will be continued by Dr. Adriaans. He is affiliated with the Maastricht Center for Systems Biology at Maastricht University, and I'd like to gladly give him the opportunity to discuss with you.
All right, thank you, Mr. Prorector. Let me start by congratulating you on a very nice thesis. I was especially impressed by the breadth of the technology and the approaches that you've used and also mastered. And secondly, I want to congratulate you on your healthy outlook on life and working together in general, which is highlighted in the proposition 7, 8, 9, and 10. I won't ask the paranymphs to actually mention them all, but congratulations to you on staying so positive. All right. So I'm a bioinformatician by training, so I'm going to put on my bioinformatician's hat. And then I arrive at chapter 4, and here you looked at expression differences in monocytes between male and female subjects. And you used gene expression microarrays for this from the Illumina platform, if I'm not mistaken. And one of the steps that you always need to do is indeed filter out lowly expressed genes or filter out background genes. And when I see here, then you chose a specific threshold of 7.5. And so my first question is very simple. How did you get to that specific threshold?
STEAM is a component. Thanks for your question. So actually, for the macroarray data, we always assume that it's a Gaussian distribution. So when I have a look at the distribution of the macroarray, we will see some low intensity. They are completely different from this another Gaussian distribution. So we just set a threshold, between that two distribution and cut out the low intensity genes.
So it's really based on assessing the distribution of the data and then
exactly.
Based on this.
Yeah.
Okay, that's really neat. Are you afraid that you would then miss out on interesting biology because there might be some, lowly expressed genes that you cut out that you might not be able to analyze or say anything about anymore?
Thanks for your question. Yeah, indeed. I assume if I there is always a risk if I fill out the low intensity genes. But if I add this in, then the results for the differential gene analysis will be not accurate. So what I can come up with to deal with this problem is we can check another independent cohort with a similar situation, similar disease, and they may have other genes which are not included in our cohort. Then we can check that gene's expression.
Okay. Yes, very clear. Do you think that using, for instance, RNA sequencing would help in this regard as well, or?
Yes, I think I have read a literature which says the RNA sequencing and the microarray, they basically can give you the similar results, similar gene expressions. Just the microarray can only test some known predefined genes, but RNA-seq they can measure more genes. So if I just use the genes which are same as the microarray in our study, I assume the results will be the same or similar at least.
All right. Yes. Thank you very much. Very clear answer. Then with regards to finding differentially expressed genes, you use LIMA, I assume, so linear modeling on these different data sets. And I noticed that you only correct for HM batch. What's the reason not to correct for any other factors in your analysis?
You mean the batch infection part? Yeah. So we correct the batch infected because we do see there are several batches in our data set, and we also have a document to record the reason, which is because they are tested from different date. So that's why they have the batch, and we correct it based on these batches. Yeah.
Mm-hmm. And if I look at table 2 in chapter 4, that's on page 128, and I also noticed that if you compare the male and female cohort, there's two differences in HDL, I think, and in creatinine, which show a significant difference between the group. What's your opinion? Should you then take these along in your corrections as well? So similar as you would correct for batch effect, should you correct for differences in level of HDL in your differential gene expression analysis?
Thanks for your question. So for this one, for the table 2, actually you can see there is basically no difference between female and males except for the creatinine. You mean table 2? So and HDL, yeah. So, but one thing is there must be some difference between female and male. Female and male, because they have their sexes, so there must be a lot of factors that are different. So it is, for me, it is not wise to exclude all of these factors. So what if we don't know if they will influence our results or not. So what we did is just because we already know the ages is indeed influence the cardiovascular disease. So we exclude this variant. For the others, we didn't do that.
Okay, yeah, very clear answer indeed. If you want to be able to find some biology in the end, it might sometimes be prudent not to correct for everything, because then how will you interpret the results once you have them? So I fully agree with you there. Then do I have time for one final question? Then in a more general sense, if I look at chapter 4 and 5, they're very much focused on the transcriptome, and that's a very comprehensive way to get a view on the potential underlying mechanisms in these particular complex diseases. Can you think of, in the context of multi-omics, because you mentioned this in your introduction as well, what type of omics data in this context, so when looking at gene regulation, what would be of added value in these types of chapters next to the transcriptome?
thanks for your question. So for chapter 4 and 5, currently we only have transcriptomics data. So if we only have transcriptomics data, if we would like to know more like a signaling pathway based on this, what we did is we can borrow the knowledge from prior knowledge resource, such as what you know, the KGG or any protein resource, which are connected to the genes, or any pathways connected to the genes. Then we can infer this activity, protein's activity, or signaling pathways activity based on this gene expression. But if we also have the protein levels information from the same patients, so we could even do like a network, because this network also already have the upstream and downstream information. So we could check the whole pathway, how this gene's expression, like how these proteins influence this gene expression, and what kind of like the middle proteins or transcription factors are. So that is what I can come up with.
Okay, yeah, very clear. Do you have time for one short final question?
We have to stop now. I'd like to give full opportunity also to the last opponent, Dr. Donners, who is affiliated with the Department of Pathology at Maastricht University, and which is the same department in which also the candidate was affiliated with. So I'd like to give you the final opportunity.
Thank you very much. So, dear candidate, I also would like to congratulate you, but also your promotion team with this beautiful piece of work. And I must admit that as a biologist, all these computational methods are highly advanced. Congratulations. Before I would like to ask my question, I would like to ask one of the paranymphs to read up the propositions number 6 and 10.
Basically, we can ask both paranymphs to each do one assessment. Yes, you can make your choice.
Proposition number 6. When the outcome of a particular bioinformatics analysis is disappointing, blindly switching to another package is not going to solve the problem. You should rather go back to check your data and dissect the approaches you currently used.
Thank you very much, and that was pretty fast, I must confess. So you could do it more slowly and relaxed. There's no hurry.
So proposition number 10, if something is too difficult to accomplish, you could just give it up and move on to another goal.
Thank you. And now the candidate.
Yeah, thank you. So I must say that at first glance, when looking at these two propositions, I thought, well, this is a bit contradictory. On the one hand, you advise to give up and move to another goal, while on the other hand, you say, well, you should go back to your data and the approaches that you use. But perhaps you can explain a little bit more what you meant with these two propositions.
Esteemed opponent, thanks for your kind word and question. Yeah. So for the number 6, actually, what I mean is, you know, there are a huge number of packages to deal with the same problem, probably like the differential gene expression or the gene cell clusters. So you basically couldn't know which one is better. So when you get like a little bit weird results, so according to my experience, it's not wise to just, because there are a huge number of methods you can use, it's not wise to just change it to another one, but go back to check your data, this distribution. Like, is there any outliers, or is there any, you should do any like data cleaning things. There must be some reasons you get these results. And for the chapter 10, actually, it's based on my experience during my PhD study. So for example, in the chapter 3, so you see actually I did the like the liver compartment segmentation without the HND staining image or histological imaging. That is because we do not have that histological image. Those images are damaged during the experiment. So in that case, I mean, we cannot ask the biologists that will take a long time to redo the whole experiment. So that's why we changed the direction. We just identify the liver compartment without the histological image. So that will, that is what I want to say in the number 10 last sentence.
So if I understand correctly, you say that in proposition 6, you have a lot of opportunities and you shouldn't blindly switch to different packages, but you're guided in principle by the data set that you have and the research question that you have, but you might be able to switch to different approaches that still can answer that research question, right? Because it's not simply switching to another package unless the research question and your data set will allow it, right?
Yeah, exactly. Unless you have that request, your purpose is you want to do that. You have that purpose, then you could find a suitable approach to deal with that problem. For others packages, they basically deal with the similar functions. Then you could better to check back to your data.
Yeah, I can only agree to that. Then another question I have. So you mentioned in your thesis also various different types of omics, and you talk about the integration of different omics techniques. And obviously, ideally, you would like to do all these approaches yourself on the same samples, the same data set. But we often have to use publicly available data sets because we don't live in the ideal world that we have all the money and all the time to do this all ourselves. But they are published by different groups and they obviously come with major challenges. So my question is, what do you consider to be the major challenges and what would be your recommendations to biologists to solve these issues when you want to integrate multiple omics techniques and data?
Thanks for your question. So for the omics integration, there are like two types. One is you have the same samples, then you could integrate the omics data by samples. So you could integrate the proteins with genes. And another way is you have the same genes. You could integrate, by genes, you could integrate samples. So there are two data integration way. So for the, if you integrate genes with other the proteins and the omics, actually you could, it's, according to my experience, it's not simply to just combine them together because proteins and genes, they have the different, they are different thing and they have different functions. So it's better to find a network. So how, when you integrate it, how the proteins influence that genes expression and how the metabolites influence the proteins and then the genes. So in this way, and we can also combine with the knowledges we already know. So there are a lot of public database we already know. So based on this, it should be a better way to integrate the multi-omics data.
Dear Ms. Candidate, you are allowed to finish your answer, or if you say, I was complete, then we just stop.
I would like to say 2 sentences. Yeah.
Okay.
So for another way, it's about the sample integration. For that one, it's just like a batch correction. So if they have the same genes and they have the same conditions, you could just use the batch correction similar way to combine the cells or the samples together. So that's my answer. Okay.
Thank you very much. Dear Ms. Candidate, the time appointed for defending your thesis has now passed. The degree committee will now withdraw to discuss not so much the quality of your thesis, but in particularly the way you have defended your thesis this afternoon. I request that you and your company await the results of our deliberations and our return to this room. Thank you very much.
Thank you.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. Southside, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. Years can't fly by now, don't wait. waste all your time. 'Cause I'll go the extra mile.
Dear Ms. Lu, the degree committee here present has assessed the quality of your thesis and in particular the way you have defended your thesis this afternoon. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the title of doctor. Professor Biesen is authorized to confer upon you this academic distinction in accordance with Dutch university customs, and I invite your supervisor now to take the floor.
Please all rise.
Dear Dr. Chang Lu, do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Chang Lu, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university. Let's see if this will not be a mess again. Would it be the first time? Congratulations. So, it's recorded.
Please be seated.
Dear Dr. Lu, dear Chang, it's a great pleasure to congratulate you with this achievement. In one, on better than 3 words, very well deserved. We both know how hard you worked for this achievement, and it's really well deserved. Also would like to congratulate your parents first, because I deeply regret that I cannot be here at your thesis event, at this highlight, possibly one of the highlights in your life. But I'm sure they must be at least equally proud of your performance and achievement today as me. Today also marks the end of our 5 years of gratifying collaboration. At least it was a pleasure from my side. In September 2007, you arrived in Maastricht in our lab to orbit in front of my room almost for the whole morning, waiting for me. For whatever reason, although I can think of at least one, I was not there to welcome you and accommodate you. Unfortunately, Mother Lieve took care of you at some point, and it took me 5 years to make up for this inelegant welcome. And I hope I succeeded in this. So in Maastricht, there were several hurdles to take for you, as you were not only exposed to a new culture, but also had to make sense of English, and above all, of the reasoning of simple biologists like me. But you readily adapted to all of this. And as we have seen today, and especially have seen the past 4 or 5 years, you can by now bluff your way into biology to some extent. The journey as a CC/PhD was remarkably smooth in the first 1.5 years. And you were able to produce almost the complete pipeline for the plaque macrophage mapping study, so the project that you were on. And that was an exception, because that is not what usually happens for PhDs under my supervisions, I can admit. Your results were looking very promising. So that gave hope. And you were already just trying to write down the first parts of the manuscript. And then the polishing started. And the polishing took almost 2 years, including the revisions. Numerous times you surprised your co-supervisor, Dr. Goossens, with entirely new outcomes and tons of pie diagrams after just minor tweaking of the settings. And that made us a bit wondering about how reliable it all was, but in the end it worked out really well and robust. For you, with the pipeline ready, this was just minutes of work. For Peter, unfortunately, it took nights, weeks, months to biologically validate your findings. But fortunately, to compensate for that, we from our side thought of increasingly sophisticated site analysis, at least from a biological point of view, that took you weeks or months to understand the underlying mathematics and to apply these tools. But it all was worth the effort, as it appears by now. By the end of year three, you got nervous on your research progress, and you had some reason to. There was a single, still unpublished paper, a few very initial contours of what might eventually might evolve into nice studies, and on top of that, the second envisioned landmark study on liver was a complete failure, as all tissues were randomized during analysis. Even worse, we had the worst pandemic since 1918, not to mention the Black Death 600 years before, and the whole lab turned into a hibernation for months. For us, it was just inconvenient, but we had still our family, close friends nearby. For you, at 10,000 kilometers distance from your family, it must be much worse. In fact, in the past 4 years, I recall you only have once been able to visit your family due to all the lockdowns and travel hurdles that have been sort of raised since then. So this illustrates again your perseverance and strength to sort of keep on going. So I admire that. Altogether, the associated stresses had serious physical repercussions that even further increased the pressure on you because you would like to finish, to get your PhD at some point. Your desk became packed with pain, stress relievers, antibiotics, advanced ergonomics, where it formerly had been packed with alien cookies, low-carb diet pills, and exercise instructions. So that already indicated that you had to combat that as well. But you overcame these hurdles, published the plaque study in a very prestigious journal, and finalized and submitted 2 other studies by now. Now, here you are, up for next step in your career, in a true bioinformatics environment with just 1 or 2 scattered biologists. So that's a much better atmosphere for you to further develop. People with whom you really can communicate about algorithms and that really matter. In Heidelberg, which some would see as the German Maastricht. As I realized, we have quite some things in common. Maybe not age or gender or size, but we both consider organized chaos as a key step to new ideas. You are the first to beat me in late delivery of a thesis. We've seen that yesterday. We both suffer from eclectic amnesia from time to time. Happily, I was not the only one, forgetting trousers and shoes after workout in the gym. Your dumplings are almost equally good as mine, and for long, I was the only one not to recognize key people at key events. And I'm just referring to the Christmas party of pathology in year 2. But please don't end up like me in the end. That's just a warning. Then a personal note. I can admit here and now that I have a weak spot for your Chinese, the 2 Chinese PhD students that have entered our lab. You have this disarming and considerate, especially when I suffered this bike accident. You were there with flowers and things together with Han. You have this disarming way of communicating, and always with a smile. So that I will miss, and that we will miss. Let me then end by wishing you the best of success and strength in Heidelberg, and I hope it will be the stepping stone for a steep career in China. You deserve it, and you have the qualities for that. I'll like to give back the word to the pro-rector at the end of my talk.
But first we applaud. So, Dr. Lu, how does that feel?
Yeah, feel nice.
Feel nice. Very good. So also on behalf of Maastricht University and our College of Deans, I would like to congratulate you. And in my congratulations, of course, I also bring forward your family, who most likely is online or maybe recorded later, and so they can look at it online. I also include in my congratulations the promotion team, who took you over the last 5 years in this journey. I know you are a CSC student, so it's always nice to see how the CSC students are doing and have finished a PhD degree. I also would like to thank the members of the Corona who took the time to join us to prepare questions. I always say sometimes it takes 7 minutes to ask questions, but it takes more than 7 minutes to make questions. That's a long time experience. I also would like to thank the audience who likes to join us here. And by that, I'm going to officially close the ceremony using my hammer, as you can see over here. And how are we going to continue? Well, we're going to make some pictures here with our member of the on-site, no, online. So Professor Olaf, you will be here, and we're going to make a picture with you. Thereafter, we're going to make with the candidate, with the young doctor, I should say. And then there is an opportunity to congratulate the new doctor in the old library. And I would like to ask all of you here in the audience already, go to the old library and we will follow you shortly. Thank you very much.
Thank you.